that's a good in some respects, because you could utilize the services if you're interested yourself. So again, the main focus of really my interest lies in trying to build that connection between functional proteomics. You know, we often hear about functional genomics, but you know, what does that really mean? If a messenger RNA is there, it doesn't necessarily mean that the protein is present or active, even if it is present. So from the standpoint of industry, genes are not drug targets. There may be some desire to eventually do gene therapy, but in reality, the pharmaceutical industry is really interested in defined protein targets that it can manipulate. And that's our best chance to treat a lot of these diseases with unmet diseases. And phosphorylation is a great way to go, because really at the end of the day, whether you're looking at different classes of cytokines or receptors, we think of over 1 million sites. So this is very problematic when we're talking about mass spectrometry. In fact, mass spectrometry, I think, is a great tool for discovery of modifications of proteins and identifying phosphorylation sites. What's very interesting is that when you start to really look carefully at the data by mass spec, and this is probably best encapsulated in the Phosphocyte Plus database that Peter Hornbeck has really pioneered. Peter actually used to work for me in my first company, and then he went to Cell Signaling Technology and started Phosphocyte Plus. It's an excellent website. It's a very good companion phosphorylation site database. Goes on really well with Connexus. We have, with our PhosphoNet site, there's a little bit of overlap, but in fact, there's a lot of things that we do differently, and they don't really have predictive data. We have predictive data in our database. So anyways, with these phosphorylation sites, when you look at the Phosphocyte Plus by mass spec, I would say about 98% of those phosphorylation sites have only been described once or twice. Very few of them do you see, like the MAP kinase, with over 2,000 reports of phosphorylation of the T and the Y, the threonine 202 and tyrosine 204, and ERK1, for example. So most of the phosphorylations, when you go by mass spec, it's a powerful tool. You can detect these, you can discover novel sites. It's very hard to track them from sample to sample. If you were to do a study by mass spec, typically where you want to track thousands of phosphorylation sites, and you want to do this in some sort of reliable fashion, you need about 20 to 30 milligrams of cell lysate protein. And you have to do your enrichments, and then you have to do multiple runs on the mass spec, probably in triplicate to get statistically significant data. You're looking at probably $50,000 plus study, probably several months to get it done properly. And you have to do isotope labeling of your cells, and that's expensive. And so, to me, it's just not a tool that, if you want to do large-scale analyses of many samples, you really have to use it as a way to identify the proteins that might be interesting, and then you have to go to, say, antibody-type technology to follow up. So, is there a way that we can use antibody microarrays as a way of tracking large numbers of proteins of interest that are low abundance in a directed fashion? And this is what we can do. So this is really contingent on the availability of really good antibodies. And so we've over the last 17 years at Connexus, and before that, my first company, Kinetech Pharmaceuticals, we developed a lot of the early antibodies for upstate UBI and stress gene. But I thought when we started Connexus that, okay, well, there's lots of antibodies out there, and in fact, there's over 1 million antibodies that are available on the market today. Now, we've tested pretty close to over 6,000 of those antibodies, and I can tell you that about 75 to 80% of them are either impotent or they're non-specific, or they just outright don't work. That's the success rate. And this is why we've had a lot of issues with reproducibility of data, and misinterpretation of data because the antibody probes aren't as good. So we've actually, in the last 2 years, started to make a lot of our own antibodies. But we've cherry-picked through about 1,500 of the antibodies from ourselves and from other companies that we now use in our antibody microarray services. Many of these antibodies will become commercially available from Conexus in the coming weeks. And so we cherry-picked them from about 26 different vendors and ourselves. Again, we're looking at low abundance proteins, and they offer some opportunity to quantify the levels of those proteins and phosphorylation at specific sites of interest that have been shown to be regulatory. So we think that the gold standard for quantifying proteins is really Western blotting. So we tried to develop a technique where we could multi-blot and maybe on a little SDS page gel, almost like a 2D gel in a way, except one dimension is immunoreactivity, the other dimension is size. We're able to look at maybe 30 to 40 proteins at a time. And we use that as a validating technology. But the microarray is where we can look at large numbers of samples where we can investigate at this point about 878 antibodies at a time on a microarray platform. And that's what I'm going to be presenting. So with this antibody microarray, it's one of various services that we have. And this is actually an image of a CAM880 chip. This was the predecessor of the chips which are now just being released from Conexus. This was our one chip that had a mix of about 600 pan-specific antibodies and around 200 or 318 or so phospho site antibodies. And these are printed in a pattern where you notice is that the duplicates are in the same grid, but not right next to each other. And we do that in case there's any distortions, dust or whatever, not to get the duplicates next to each other. You can see there's a fairly consistent pattern. And the more red the signal, the stronger the signal, and blue is the weakest signal, and yellow and green is sort of in between. And we can take these images and focus in on one little part of the grid and take a control sample, colorize it blue, for example, and take a treatment sample, colorize it red, and then overlay the two grids. And then whatever white is unchanged, and whatever blue has gone down, and whatever red has gone up. So we can kind of visualize that here, where we can see changes in response to, in this case, EGF treatment of A431 cells that overexpress EGF receptor for 5 minutes after they've been serum starved overnight. And we can quantify these, of course, and we can do this with about 3 orders of magnitude and variation. So antibody microarrays are extremely difficult to work with. The standard procedures that people have used have been highly flawed. A number of companies offered antibody microarrays. Thermo Fisher was the first. Clonetech came up with one. Sigma had one. Full Moon still has one. We have ours. We've been doing this now for about 7 years. All of these antibody microarrays are now off the market, with the exception of the Full Moon one and ours. And that's because the data from the microarrays is very difficult to reproduce by Western blotting. Often the antibodies aren't available to even do those experiments. It's very expensive. And it turns out that the way they were doing it was actually fraught with technical difficulties. So I'll explain that in a moment. But when we print these spots and we look at the signals that we get, the median value of the variation from the average of the spots is about 12%. So actually it means that you can see some significant changes, even 40, 50% changes are probably real. And we have over 3,000-fold dynamic range. We can have counts that are accurate with duplicates down around 50 counts and go up to even 200,000 counts or higher. So it's a very amazing dynamic range. So we are just about to launch 2 new chips. One is the CAM900 P chip for phospho, and this has 620 different phosphotype antibodies on it. And it has, for the same phosphoproteins, 258 pan-specific antibodies. So this allows you to look for changes in phosphorylation of some of these signaling proteins, mostly kinases, and the levels of those proteins. So now you can see, well, is that increase in signal because the total amount of the protein has gone up, but the stoichiometry hasn't changed? Or is it because the level of the protein's unchanged and the stoichiometry has increased? This is always a problem, especially if you're doing studies like mass spec, for example, when you see increased phosphorylation. Is that because the protein changed, or is it because the phosphorylation of the protein is altered? So we can address that now. And the 900E chip for expression, it has 878 pan-specific antibodies for protein expression. And so I'll be showing you some new approaches that I think that we're looking at disruptive technology here. So if you use the two chips together, you have coverage with about 1,500 different antibodies now, which is greater than anyone else on the market ever in the past, nor anything I've seen in publications. So that is now almost available. We just received the printed chips, in fact, today. So now we're doing the final test. So the basic concept here is that you take a protein, you dye label the protein with a fluorescent dye, and one of the tricks that we use is we use a combination of dyes, because as I was mentioning earlier in our discussions, different proteins bind different dyes differentially. So we get a more complete labeling of proteins if we actually use a blend of dyes that fluoresce at the same wavelength. And then afterwards, this fluorescently tagged proteins in your crude cell lysates is incubated with your microarray, and it will bind. And what you're doing is you're quantitating the signal of that captured protein, and that's what you see as spots. And that's typically the way it would be done by most microarrays in the past. And in fact, what some companies have done is much like dye GE, where you dye label your control and your treatment sample and you incubate them together on the microarray, then you measure your wavelengths for the control sample and the treatment sample at different wavelengths, and then you look for changes. We know that in fact 80% of the changes we see on a microarray like that are dye-related artifacts of differential dye binding. Nothing to do with the change in the level of protein.
But you can swap it, right?
That's when you swap it, that's what you see. The data is not reproducible.
Even you swap?
When you swap, yes. On the microarray, we can see that the captured proteins have most of the changes that we're seeing. When you swap the dye, they're not reproducible in the same directions. So we know that this is a big problem. So one of the designs of our chips is we have two fields printed on the same chip, separately incubated, the control sample, the treatment sample, and each of them has two antibodies measurements per field. So essentially each antibody is printed in quadruplicate. In the two fields, duplicate measurements are made. And we use the same dye labeling for both samples, and they're on the same chip, and they're all incubated. The only difference is that the samples are separated. And that gives us the most reproducible data, and we avoid the dye-related artifacts altogether.
So, sorry for that.
Yeah, please go ahead. It's fine to interrupt. Any questions?
So you said that only one chip you incubate with two samples?
That's right.
How do you do that?
We have a chamber, a little
Separate
gasket
chamber.
that separates the actual sample. We can load about 400 microliters of volume in each chamber. Yeah.
So in these chips, you mentioned we have several of them. You can measure the pan-phosphorylated proteins, you can measure the phosphorylated proteins. What you have is just presence or absence, increase or decrease. So you don't have quantification of the intensity of the change or of the increase.
Well, they're relative to each other. So what you do is you take the signal for your treatment and your control, and you then calculate from the signals the percent change from the control. And then you measure your error in your signals, and you sum up the errors, and that gives you a good sense of what if your percent change from control is 100%, and the sum of your errors is 30%, you have a higher confidence. That's a real
So
change.
you can quantify the change.
Yes,
Okay.
absolutely. We quantify every spot.
Two questions. One is that when you do this kind of a quantification, like in a normal microarray, if
Yes, we have a normalization protocol. It's a very good question. Our assumption is the null hypothesis, that if you have two samples and you've undergone a perturbation, as long as it's the same model system, that there is no change. And so what we do is we sum the intensity of all the signals in each field for every spot, and we expect that the sum of the two fields will be the same. And then what happens is, if you do that, the worst case scenario is you make changes not as dramatic as they actually are, but you don't make changes appear greater than they actually are. So we normalize all the data. We have the raw data, but we also normalize, and then we calculate the averages and the percent change from control, and calculate z-scores.
And that is therefore a global normalization.
That's right.
And my second question is that, let's say, since it is an antibody adjuvant, so now, have you tried to see for this each antibody, how do they react with isoforms of a particular protein?
That's part of the characterization that we actually are doing, and we've been trying to develop with our own line of antibodies, isoform-specific antibodies. So we have antibodies that pick up ERK1 but not ERK2, or AKT1 but not AKT2 or AKT3, and that's what we've been developing.
So what's the process you actually develop?
We get recombinant purified protein kinases, and on dot plots we test spotted with the actual purified kinases. We probe them with the same antibody, and we expect to see a signal against the target kinase, but not the other isoforms.
Do you try to mutate them, make a mutated clone, and then see that whether really it is specific?
We have access to mutated kinases. Currently we have about 400 different kinases that we have access to, and then there's mutant forms about another 100 of those. So we can do that, and that's how we're testing a lot of our antibodies. Against purified kinases, against dot plots, with phosphocytes, we actually have multiple peptides, because we do peptide synthesis in-house, with the phosphorylation sites and related phosphocytes, and then we test the phosphoantibodies against those related phosphopeptides, and then try to epitope map them. So we get a sense of what its overlap is.
So for making a specific antibody for a specific isoform, do you actually predict based on these in silico kind of epitope mapping programs, or how do you do it?
We actually test it. We put those phosphocytes onto dot plots, and then we test it against the various antibodies.
Peptide that you synthesize.
That's right, against the peptide.
And before you do that, you possibly do a bit of mapping, which is kind of a computational. Do you do that or no?
We're restricted in part. We can do spot synthesis, so we could do all kinds of variations, but we tend to use purified peptides by multi-pep synthesis that at least are around 80 to 90% pure. So that's what we're using.
But when you do this thing, you are doing your whole thing based on a particular peptide. But now, when you normally take a cellular extract, does conformation of the protein also matters when it binds to this?
It's a very good question. Let me show you the next step, and I think I'll answer your question. The problem with using intact proteins that are dye-labeled is that you may have a situation where it's a monomer and it's captured by your antibody. Target A, is binding to antibody for target A, and it's a monomer, and your signal is reflecting that protein. But most proteins inside cells are complex with other proteins. So in fact, what you really can often have is a situation where the target protein is binding and has dye signal, but associated proteins are also binding, and they contribute to the dye signal. And what you're really looking at, where there's a change in the signal, is a change in protein-protein interaction. Not the level of the target protein, nor its phosphorylation, but this interaction. The other problem is that some antibodies, we found the Fc portion can actually bind proteins and peptides. It really threw me off when we started doing peptide analysis with antibodies and dot plots, and we started finding that certain peptides were binding to all of our antibodies. And so the Fc portion of antibodies actually does have binding affinity, in addition to the variable region. The other thing is that you can often have a cross-reactive protein, something that shares an epitope or it's very similar enough that at high concentration this protein will now bind. So you get false positives that way. And then you'll also get false negatives if the epitope that's involved in the interaction is binding another protein and it's blocked from binding. So these are all issues with antibody microarrays. It's other major issue, it's a two-edged sword. The antibody microarrays, I would, based on the data that we have, is about, I would say, at least 50 to 100 times more sensitive than Western blotting. So often we can see a lot of results in the microarray, and we cannot see any bands on Western blots. And it makes sense because you're really concentrating the antibody and you're concentrating the antigen. And so you can see those interactions that you just cannot see by Western blotting. So it means it's hard to validate sometimes the results by Western blotting. Now, to get around this problem, what we've developed is a chemical cleavage procedure. So instead of starting with a dilabeled protein, we do a chemical cleavage and we cut at cysteine residues. We don't want fragments to be too short, and we definitely want to stay away from things like arginine and lysine residues because a lot of phosphorylation sites are flanked by those kind of residues. So we cut at cysteine residues, and what this means is a protein like mTOR, we can get an equivalent dye labeling of the fragment we're capturing as we would get with the RAS protein. Because often, when you do, for example, a dye GE labeling, the proteins that give you the really nice strong signals are going to be the big ones because they can bind more dye. They may be low abundance, but if they're structural proteins, then you've got big structural proteins with lots of dye, and the proteins that are small, you can't see because you can't get as much dye labeling. Here, with the chemical cleavage, we're essentially taking all the various proteins and getting a sort of bell-shaped curve of an average size fragment, which we then dye label. And then that fragment that contains the epitope will then be incubated with the array and it will bind. So we get a more consistent degree of labeling amongst proteins of very different size. And because we fragmented it, we get rid of a lot of the protein-protein interactions. So more of what you see is a reflection of the target protein or its phosphorylation.
So do you, in your experience, does the KD of the antibody and the peptide relatively the same?
I wish it was. And the antibody KDs vary dramatically. We try to adjust for it by the level of the antibody that we put onto the array, because we don't want to have a super high affinity antibody for a protein that's in high abundance. So now we get a signal super strong, and then the scanner kind of gets confused, and then it ignores the weaker signals. That's why we're still able to, this is really good dynamic range. We can get very weak signals. The way we deal with this problem is that for all of the antibodies that are on the microarray, we run them through a series of different types of lysates, and then we calculate and provide as part of our reports the lowest signal ever recorded, the highest signal recorded, the median signal, the average signal. So you get a sense that when you get a signal back for your lysates, is this really a strong signal for that particular antibody, or is that as good as it gets? Yeah. So this configuration, here we can see the effect of chemical cleavage. Here's what it looks like from A431 cells using the standard technique without chemical cleavage. If we do the chemical cleavage, you'll notice the spots are dramatically reduced. In fact, if we overlay the two, everything that's blue there is where the signal intensity has actually been decreased because the chemical cleavage has resulted in loss of those signals. But what's white is unchanged. And there's a few red spots, tiny ones, but occasionally you expose an epitope a little bit better when you do the chemical cleavage. So that's what we tend to do now, and we recommend that for measuring the levels of expression or phosphorylation of proteins using the standard, we call it Kinex antibody microarray, the CAM type analysis. So we can see changes in a signal from A431 cells, lots of spots that light up. And then when we overlay, we can see nice changes of what's gone down and what's gone up with these kind of treatments. And they're each quantified. So that's state of the art of what's available in the market today and what's been described. Where we're going now is sandwich antibody microarrays. And so if you Google the terms sandwich antibody microarray, you can hear a few hits. Largely because it's been very hard to even get antibody microarrays. So the sandwich antibody microarray, SAM format, what we do is we have our target protein. We don't do the chemical cleavage here because the purpose of this approach is to look at total changes in phosphorylation. So we don't want to fragment off other phosphorylation sites. So here we have our protein, we incubate it with the array, just the crude cell lysate. And by the way, we typically use about 50 micrograms of protein to do an analysis from crude cell lysate. We can take tissue, we can take extracts from cells, we can take serum, we can take urine. We've gotten samples of all of them that work on the microarray. So it's extremely sensitive. So we capture the protein, and now what we do, let's say this is a tyrosine phosphorylation site, we incubate it with a reporter antibody where we've dye-labeled the antibody. And so this particular antibody, I have a little bit of information about it, see if anybody wants a copy. It's called PYK. It's a polyclonal, generic polyclonal antibody that's generated against hundreds of different phosphorylation sites and then affinity purified over phosphotyrosine agarose columns. And it's much better than any of the monoclonals that are available. When you look at the report there, you'll see that the monoclonals tend to favor, well, acidic amino acids surrounding phosphorylation sites are negative determinants for these antibodies, but not for this antibody that we have here. So we also run this antibody down an IgG column to get rid of any non-specific interaction with IgG. And it's dye-labeled, and then basically we incubate it, and we record the fluorescent signal that's associated with that spot. We know what the kinds of or protein is captured, so if we see an increase in signal, then it's tyrosine phosphorylated. So here's an example of what a CAM-A80 chip looks like for cells that have been treated with EGF. We get lots of signals from tyrosine phosphorylated proteins, and they undergo changes with the EGF treatment. Well, you can also use the SAM approach to look at specific protein-protein interactions. So you have a situation where you have adapter proteins, GRB2, all these proteins that associate with receptors that can feature SH2 domains or PTP domains, and you want to know what proteins are they interacting with, and how are those interactions changing as a consequence of your model system. So with this particular approach, we can actually heat shock proteins, 14-3-3 proteins that are looking at phosphorylation interactions. You can look to see what proteins might be targeting by a particular adapter or chaperone protein that are in a model system. So here we capture the complex, and then what we do is we incubate with the reporter antibody for the other protein of interest. And then we quantify the signals for that. So this is a way to actually look at large-scale changes in protein association with specific adapter proteins. So I don't have any data to show you results of that right now. Now that we have our new chips, this is where we want to get more of this data available before we start to offer it as a service. Now, we have another approach to quantify phosphorylation. I told you with the PYK antibody, with the sandwich assay, you look at tyrosine phosphorylation. But another approach is to actually use a dye, which is called Piamago, which is developed by a company called Tamora, which is in Indiana at Purdue University. And we've been working with them over the last year and a half to optimize the system for detection with microarrays. And the Tamora reagent, it binds serine, threonine, and tyrosine. It's a nanoparticle. It's much more sensitive and more uniform in its binding than ProQ-diamine, for example, which is often used in industry. And so you capture your phosphoprotein, and then the Piamago reagent binds, and this Piamago reagent is biotinylated. And so then you detect with a reporter antibody, this antibiotin antibody. And this has been the best combination that we found that works for us. Now I have some data with an alternative approach where the P-Imago reagent is actually directly fluorescently tagged, and you can buy that from Timora. And so I have data here, the results of C-star, where we have total phosphorylation signals in a maturing starfish oocyte. But now that we found that the antibiotin antibody approach is much better than the avidin approach. We're finding that avidin binds a lot more non-specifically to proteins on these arrays, and we get much cleaner results if we stick with the antibiotin antibody. So that's what we're using now. And then another approach, and we talked a little bit earlier about functional assays. One of the probes, and that previous array was the PAM array for the P-Imago antibody microarray. This is the FAM array. This is for FSBA. This is an ATP analog that covalently attaches to the lysine residues in the active site of protein kinases, only if the active site is open. So this ATP probe, basically we capture the kinases in the array. There's over antibodies for over 400 kinases on the array. And then what you do is you incubate it at the ATP binding site with this probe, FSBA, which is biotinylate. And we make this probe. And once it binds, then you have, again, an antibiotin reporter antibody, and it binds. And this gives you a sense of what protein kinases have the ATP binding site in open conformation. In a sense, what might be the activated forms of these kinases. And so here, again, I've done it with the avidin biotin combination. Again, I would recommend going with the antibody for biotin that's dilabeled instead. It's more sensitive. But you can see here, even with the avidin, we get nice signals for detection of proteins on these arrays. Now, there will be obviously some cross-reactivities. There will be some non-specific results, but we can then go back and check that. So one of the things that this approach with the FSBA can be adapted to is counter-screening for kinase inhibitors. So what you can do is you can capture the kinase like before and have the kinase inhibitor incubated, and then you add your FSBA probe. If many of these kinase inhibitors are ATP analogs, then they block the active site. So the probe will not bind, and therefore you won't get binding of your fluorescent dyes. So for those signals that are reduced in the presence of the inhibitor, they're likely potential targets. Now, as we've explained earlier, there's about 1,300 diseases at least involving kinase defects, and the pharmaceutical industry is producing a lot of inhibitors. The FDA has already approved at least 27 of these, lots more in development. We have all of these drugs here, these inhibitors that are available from these companies that have been released that target all these various kinases. They may very well be also working on other proteins, or maybe analogs of these with slight variation may be more specific for some other targets. So we can use these kind of drugs in this format. Now, I think I've already shown you that while oncology has the bulk of the activity right now, Alzheimer's disease, abnormalities, tau protein, Parkinson's disease, alpha-synuclein problems, just dealing with the people with the Huntington disease, where there again seems to be phosphorylation defects along with the repeats of the glutamine. And that's another story, actually. Maybe around the dinner table I can tell you about. But one of the things that we did a few years ago was we actually took purified kinases and we put them onto microarray chips. And we wanted to use these to see if we could do drug screening. So what you see here is part of an array where these kinases were printed at triplicate spots, and then they were probed with the FSBA probe. So the FSBA binds, covalently attaches, and this probe was biotinylated so we could quantify whether we got any binding. And using that approach, we did try to take drugs and on the microarray look to see with star-sporin, a very non-specific kinase inhibitor, do you get reductions of signal? This is the percent change from the untreated sample at 2 different doses of star-sporin. So you can see that actually, yes, you can interfere with the binding of the FSBA probe with drugs. Yes? What it does is, the FSBA is an ATP analog that binds to the kinase where ATP would bind, but it covalently attaches to the lysine, so it doesn't come off. And so what we're seeing is this staurosporin is an ATP analog, so it can't bind. Or sorry, it binds, and then the probe can't bind because it's being spherically hindered by the staurosporin. Well, what happens is, with the antibody microarray, you can capture your proteins on the array with the antibodies, and usually what will happen is the ATP will get washed away, depleted. So then when you go in your probe, you can get the label. I could show you earlier, we get nice labeling with the FSBA probe. This is with purified kinases. So now the advantage is that we can take crude cell lysates that have a larger repertoire of kinases. This is with 200 kinases we did this. The problem with these chips, where we never marketed them, is that the chips are unstable. Once you put the recombinant proteins on the chips, they're okay for a couple of weeks, but after long-term storage, they seem to open up and they lose their activity. But now, we can take the crude cell lysate, incubate it with a microarray to capture the endogenous kinases in their active states, and then use it right away. So we think we can get much better results. But this is showing, at least from proof of concept, that you can use different drugs with purified kinases on a microarray format using the FSBA probe and get competition. So where we're going now with this is that in principle, we have essentially about 7,000 measurements per antibody microarray. Okay. Sorry, 3,500 measurements per microarray. However, there's no reason why we can't go to a double labeling process so that we can take and start with our phosphoproteins, dye label them with one dye, incubate that with the array, scan the array chip, so we get clean measurement of that, and then incubate with a, say, a Piamago reagent. And what happens is those conditions sometimes take off a little bit of the dye. We've noticed that. But we then can go in now with the sandwich antibody approach with a different dye and get 2 measurements per spot. So now we're getting multidimensional. And because it's the same array, we can use all the various formats, essentially, detection systems, and start to multiplex the data for a particular antibody spot. So this really is, we're now talking about 7,000 measurements potentially, and we'll be going probably in a year to about a 950 antibody microarray. So we'll steadily increase that. So we use these microarrays in different applications. These are some of the earlier applications, but I like to present it because it illustrates some of the issues that we're dealing with biomarker discovery. So in Alzheimer's patients, we took the serum from Alzheimer's patients and pulled out the peripheral blood lymphocytes, and we ran them down an early microarray, one that had only about 622 antibodies. And from this clinical trial, where we had mild cognitively impaired patients and patients that had Alzheimer's followed over a 2-year period, we could completely separate the Alzheimer's patients from the mild cognitively impaired patients, shown here, male and female, in the dark circles and triangles for Alzheimer's from the controls. Now this worked because we didn't look at one biomarker. We actually looked at, in this case, each patient is a column, and a very different antibody for percent change from controls. Green was when it went down, red is when it went up relative to the average of the healthy controls. In the Alzheimer's group, and we also looked at multiple sclerosis, and myasthenia gravis, and HTLV. So T and B cell diseases, MS is kind of a mix. But with the Alzheimer's, using those biomarkers, we can completely separate the two populations. A blood test for Alzheimer's disease.
So which stage of the disease that you actually do this kind of?
They were taken at the end of that trial, where they followed them for 2 years, so they can tell which ones were the Alzheimer's and which ones were the mild cognitively impaired. It's very difficult to diagnose Alzheimer's until you're dead. They're looking for plaques and tangles, but you don't see that until you do the biopsy of the brain. So another study was done with a collaborator at UBC, University of British Columbia, where we had mice that had these defects in the presenilins, as well as the amyloid precursor protein, that with these mutations, these mice give rise to Alzheimer's disease. And these mice were treated with basically orange tricarboxylic acid, ATA, for 48 days, and then we did the profiling, because their behavior is like they're normal, even though they're genetically like Alzheimer's, and if not given the ATA, they would develop the Alzheimer's-like symptoms. So we looked at these mice using our microarrays, and in short, basically we were able to reveal changes with our antibodies in specific proteins, and that the ATA reversed the changes that we were seeing in the models. So this gives us some biomarkers. Why do we want biomarkers? Well, if you have a disease and you want to treat it, how do you know your treatment's actually working on a disease where you're losing, your memories? You can't test that easily in animal models. You need some actual biomarkers to track to see if the therapeutic activity is actually doing what you want.
How many of these phosphorylation sites specific?
About half of them.
That's a pretty large number.
Yeah.
Yeah.
It's not for Alzheimer's, just as a common question that when you look for a biomarker for a disease, protein-based, so maybe between two diseases, the same protein can show different.
That's right.
So how do you actually develop a biomarker for a specific disease?
Right. And I will get to that in one moment. So here we got ALS, just another disease showing it in basically cerebral spinal fluid. We were able to identify that there was actually normally in these patients that had ALS, the level of this protein actually drops in the cerebral spinal fluid. And we validated that by Western blotting and confirmed that this was a biomarker. And it turned out to be a protein kinase of the EPH family. So the other way to go is reverse arrays. So with reverse arrays, you can take large numbers of patient samples. Now that you've defined potential biomarkers, I mean, what we recommend is that you do a mass screen first with the antibody microarray, then you validate the result by Western blotting, and for those leads that in pooled samples seems to work, now you investigate individual samples. If you have a large number of individual samples, then you spot, rather than antibodies in the microarray, you spot the lysates from the samples separately on the microarray, and then you probe that field of lysates with one of the biomarker antibodies. And then you see, is it a biologically validated? It's not something that was just in a few samples, but is a more general trend. And that's how you can start to define those kind of biomarkers we really want. Very few biomarkers ever make it that far. A lot of promising ones. So we spot these cell lysates with different drugs and hormone treatments, different dilutions, and then we essentially what we're doing is we're spotting them individually, and then what we do is we incubate with a primary antibody for the biomarker. In this case, it'd be for a phosphorylation site on a protein. And so if the antibody binds, you wash away the unbound antibody, and then you come in with a secondary antibody, in this case, horseradish peroxidase coupled. And then with a tryptamine dye, you can look to see the level of that protein that's there. And interestingly, in this kind of a study, we've used this to look at nice curves of dose response curves, for example, with EGF concentrations, changes in time courses. I had a hell of a time dealing with A431 cells with EGF treatment. We did Western blots with antibodies that we developed for the EGF receptor tyrosine phosphorylation sites. And when we took 5 minutes treatment with EGF, we got no signals. And I thought, well, is the EGF gone wrong? Is it the cells or something happened to the cells? We looked at all kinds of possibilities, and in the end, it was because we had good EGF, we had good cells, the effect was evident in 1 minute treatment with EGF and completely reversed by 5 minutes treatment with EGF. All the tyrosine phosphorylation was almost like 90% of it was gone with 5 minutes treatment with EGF. So timing is absolutely important. So these kind of dose response curves, time courses are important. Here's a study that we did with a tobacco company. I never thought I'd be working for a tobacco company, but anyways, what they wanted to do seemed reasonable to me. They had 80 rats, 10 in each group, litter matched controls. 10 females, 10 males, separate, so sex control. And they were exposed to cigarette smoke in a chamber in a particulate for 5 days, 3 hours per day, and they had a low dose, a medium dose, and a higher dose equivalent to a chain smoker. And we analyzed them with pooled materials from the groups first with the microarray, and then we went back and validated the biomarkers, and then we did the lysate microarray. And then what I did is I arranged the signals. I don't know if you can see this from the back, but each animal is a separate bar, and these bars are oriented so that the lowest signal is on the left and the highest is on the right. So you can kind of compare the shapes of, for example, this signal here with over here with the female with more treatment. And to make a long story short, the females were far more sensitive to cigarette smoke than the males. The initial response was protective, but with medium dose, it now became deleterious. In the males, there was almost nothing happening with the low dose. In the medium dose, it was actually the protective responses were coming on, and with the high dose, then it was like the females, it was actually deleterious. So females are far more sensitive to cigarette smoke and turning on these systems. And we also can observe that, especially amongst the controls, the variation in controls could be more than twofold. So litter matched, sex matched, animals can have a twofold variation by Western blotting in their signals. So when you're seeing that twofold variation, it shows you how hard it is to actually get a good biomarker. And the data I showed you is from the examination of the lungs. We also looked at other tissues. We haven't published this yet, but we get even more dramatic changes, for example, in the aorta from animals in these groups. So basically, we see a situation where you go from a microarray, you validate by Western blotting the results from your microarray, and then you go to a lysate microarray to do the validation. And we have multi-blots where we can have panels of about 30 different antibodies utilized. Different panel of antibodies in each lane. We use a multi-blotter to separate the antibodies from each other. We run an SDS page gel with one lane down the entire gel. The multi-blotter goes on, and in the different lanes, you have different antibodies. And so we get like a 2D gel. And we can overlay these images. And so now, unlike a regular 2D gel, where you're not seeing low abundance proteins, we can see low abundance proteins and we can quantify them. And we know they're the right proteins because of the molecular weight. And what you see here is an overlay of two gels. The control gel, these are cells that are treated, Jurkat cells.